So the physiological effects of plant growth regulators. Now these are also called phytohormones or plant hormones. Now auxins. So this was first isolated from human urine, right? So it was first isolated from human urine, right? Now, now, auxin means to grow. Okay. So auxin. So auxins was first isolated from human urine. The term auxin is applied to the indole 3 acetic acid, that is IAA, indole 3 acetic acid, and to other natural and synthetic compounds having certain growth regulating properties. Now, they are generally produced by the growing apices of the stems and roots, from where they migrate to the region of their action. Right. Okay. Now, auxins like IAA, indole 3 acetic acid, and indole butyric acid, that is IBA, have been isolated from plants. So auxins like IAA and indole butyric acid have been isolated from plants. NAA, naphthalene acetic acid, and 2,4-D dichlorophenoxyacetic acid, phenoxyacetic, are synthetic auxins. So NAA and 2,4-D dichlorophenoxyacetic are synthetic auxins. Now all these auxins have been used. Now all these auxins, that is synthetic and natural, that is IAA and IBA. So all these auxins have been used extensively in agriculture and horticulture practices. Now, auxins help to initiate rooting in stem cuttings, and application widely used for plant propagation. Auxins promote flowering, that is example in pineapple, they promote flowering. Now they help to prevent fruit and leaf drop at early stages, but promote the abscission of older mature leaves and fruits. So auxins help to prevent leaf drop and fruit and leaf drop at early stages, but promote the abscission of older mature leaves and fruits. Means if the leaves and fruits are older, they promote it, they want to get it old and fall. So this is what auxin does. Now in most higher plants, the growing apical bud inhibits the growth of the lateral bud, that is axillary bud. Now a phenomena called apical dominance. Now removal of shoot tips, that is decapitation, usually results in the growth of lateral buds. Now it is widely applied in tea plantations and hedge making. Can you explain why? So for the same reason. Okay. Now you can see in the diagram that apical dominance in plants. A plant with apical bud intact, A, means apical dominance showing, so the lateral bud is not stopped, axillary bud is stopped. Now a plant with apical bud removed, that is removal of shoot tips, that is decapitation. Note the growth of lateral buds into branches after decapitation. So as you can see in the second diagram. Okay. Now auxins also induce parthenocarpy, example in tomato. They promote flowering in pineapple. As you know, auxin promote flowering in pineapple. Now auxin also induce parthenocarpy in tomatoes. Now auxins are widely used as herbicides, that is 2,4-D dichlorophenoxyacetic. It is a synthetic auxin, now widely used to kill dicot weeds, and it does not affect mature monocot plants, so it is specific in nature. No. So auxins is used to prepare weed-free lawns by gardens. So these auxins, it is used to prepare weed-free lawns by gardeners. Now auxin also controls xylem differentiation and helps in cell division. So it controls xylem differentiation and it helps in cell division. Now gibberellins. Now what are the gibberellins? Now gibberellins. So gibberellins are another kind of promotory PGR. So there are more than 100 gibberellins reported from widely different organisms such as fungi and higher plants. So gibberellins are denoted as GA1, GA2 and GA3 and so on. However, gibberellic acid GA3 was one of the first gibberellins to be discovered and remains the most intensively studied form. So all GAs are acidic, right? Now gibberellins, that is GAs, produce a wide range of physiological responses in the plants. Their ability to cause an increase in length of axis is used to increase the length of grape stalks, right? Now gibberellins cause fruits like apple to elongate and improve shape. So gibberellins cause fruits like apple to elongate and improve shapes. Okay. Now gibberellins also delay senescence, means they delay the aging, they delay the old stage. Okay, they delay the senescence. Thus the fruits, now this is why the fruits can be left on the tree longer so as to extend the market period. Now GA3 is used to speed up the malting process in the brewing industry. It means in alcohol industry it can use to speed up the malting process. Okay. Now sugarcane stores carbohydrate as sugar in their stems. Now spraying sugarcane crop with gibberellins increase the length of the stem, thus increasing the yield by as much as 20 tons per acre. Now spraying juvenile conifers with GAs hastens the maturity period, thus leading to early seed production, right? So spraying juvenile conifers with GAs hastens the maturity period, thus leading to early seed production. Now gibberellins also promotes bolting, that is internal elongation just prior to flowering, in beet, cabbage and many plants with rosette habit. So this is gibberellins. It shows bloating and it promotes speed of the malting process. Okay. Spraying juvenile conifers with GAs, early seed production, sugarcane, is all the functions of apple and okay, and the increase in the length of grape stalks. So these are all the function of the gibberellins, that is gibberellic acid, right? Now cytokinins. Cytokinins have specific effects on cytokinesis and were discovered as kinetin. They were discovered as kinetin, right? A modified form of adenine, a purine. Kinetin, a modified form of adenine, a purine. Adenine, guanine are purines. Pyrimidines are uracil, cytosine and thymine. So from the autoclaved herring sperm DNA, a kinetin does not occur naturally in plants. So search for natural substances with cytokinin-like activities lead to the isolation of zeatin from corn kernels and coconut milk. Now, since the discovery of zeatin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. Since the discovery of zeatin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. So natural cytokinins are synthesized in regions where rapid cell division occurs. For example, root apices, that is shoot apical meristem, root apical meristem, root apices, right? Not shoot. Root apices, developing shoot buds, young fruits. Now it helps to produce new leaves. Cytokinins help to produce new leaves and chloroplast in leaves, that is bound organelle. Then lateral shoot growth and adventitious shoot formation. So cytokinins also help overcome the apical dominance. that is removal of the shoot tips, that is decapitation. So cytokinins helps overcome the apical dominance. Now cytokinins also promote nutrient mobilization, which helps in the delay of the leaf senescence. Now why does it take the delay of leaf senescence? Because they promote nutrient mobilization. So until then it helps in also the delay of leaf senescence. Now ethylene. So ethylene is a simple gaseous PGR. Ethylene is synthesized in large amount by tissues undergoing senescence and ripening fruits. Now, influences of ethylene on plants include horizontal growth of seedling, swelling of the axis and apical hook formation in dicot seedling. So horizontal growth of seedlings, swelling of the axis and apical hook formation in dicot seedling. Now ethylene promotes senescence and excision of plant organs, especially of leaves and flowers. So ethylene promotes senescence and excision of plant organs, especially of leaves and flowers. Now ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruits. Okay, now this rise in rate of respiration is called the respiratory climatic. Now ethylene breaks the seed and bud dormancy and initiate germination in peanut seeds. So as you can see, ethylene can also be known as promoting PGR. So initiates germination in peanut seeds, sprouting of tomato tubers. Now ethylene promotes rapid internal petiole elongation in deep water rice plants, so that it helps leaves upper parts of the shoot to remain above the water. Now ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Now ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. Now as we know that auxin promote flowering in pineapples, but in here ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. Now ethylene also induces flowering in mango. As we can see the tomatoes, the auxins induce parthenography in tomatoes. So induces flowering in mango by ethylene. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. Which one PGR is the most widely used? Most PGR in the agriculture is not auxin, but it is ethylene. Now, the most widely used compound as source of ethylene is ethepone. Now, even after, the most usually ethylene is the compound ethepone. Now ethepone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. So ethepone hastens fruit ripening in tomatoes and apples and accelerates excision in flowers and fruits. That is thinning of cotton, cherry and walnut. It promotes female flower in cucumbers, thereby increasing the yield. So it promotes female flowers in cucumbers, thereby increasing the yield. So ethepone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. Ethepone hastens fruit ripening in tomatoes and apples and accelerates excision in flowers and fruits. In tomatoes and apples, then accelerate excision in flowers and fruits. Now the fifth one is the abscisic acid. Think the last PGR. The abscisic acid ABA was discovered for its role in regulating abscission and dormancy. Okay, when unfavorable condition reaches, the seed goes into the abscission or dormancy. But like PGRs, but like other PGRs, abscisic acid also has other wide-ranging effects on plant growth and development. Abscisic acid acts as a general plant growth inhibitor and an inhibitor of plant metabolism. So it also inhibits metabolism of plant. So ABA, abscisic acid, inhibits seed germination. It stimulates how it does. It stimulates the closure of stomata and increases the tolerance of plants to various kinds of traces. Therefore, it is called as the trace hormone because it increases the tolerance of plants to various kinds of traces. So, it is called the trace hormones. Right. Now, ABA plays an important role in seed development, maturation, and dormancy. Now, by inducing dormancy, now ABA, abscisic acid, helps seeds to withstand desiccation and other factors unfavorable for growth. Now, in most situations, ABA, abscisic acid, that is abscisic acid ABA, acts as an antagonist to GA, that is gibberellic acid. Now, differentiation and every phase of growth, differentiation and development of plants, one or the other PGR has some role to play. Such roles could be complementary or antagonistic. Okay, complementary or antagonist. This could be individualistic or synergistic. Now, all this effect suggests that for any and every phase of growth, differentiation, development of plants, or one or the other PGR has some role to play. So all these roles could be complementary or antagonistic, and this could be individualistic or synergistic, right? Now similarly, there are a number of events in life of a plant where more than one PGR interact to affect that event. Example: dormancy in seeds, birth, abscission, senescence, and apical dominance. Okay, remember the rule of PGR is of only one kind of intrinsic control. Along with genomic control and extrinsic factors, PGR play an important role in plant growth and development. Many of the extrinsic factors such as temperature and light control plant growth and development via PGR. So plant and light control plant growth development via PGR. Okay. The sum of such events could be vernalization, so temperature and light that controls the plant growth development through PGR. Okay. So vernalization, then flowering, then dormancy, then seed germination, then plant movements. We shall discuss briefly the role of light and temperature, both of them the extrinsic factors on initiation of flowering. So temperature and light. So these are the two factors that directly affects on the initiation of flowering. So this is your chapter being done. I finish your chapter. I'm going back again to see if I left something. Diagrammatic comparison of absolute and relative growth rates. Both leaves A and B have increased their area by 5% in a given time to produce A dash and B dash leaves. As you can see, Okay, so this is what I missed it. So diagrammatic representation. Okay, in your detection of zones of elongation by the parallel line technique, zones A B C D immediately behind the apex have elongated most. The ABCD, so detection of zones of elongation by the parallel line technique, zones ABCD immediately behind the apex have elongated most. So this is your chapter being done.